Hello, John Paul. It's a great pleasure to speak to you today about my Algologies project and about seaweed and its importance in microbiology. To introduce you to people listening in, John Paul is a microbiologist, a retired microbiologist, but was formerly the Public Health England lead microbiologist for the southeast of England. Um, he holds he was awarded an honorary professorship by Brighton and Sussex Medical School through Sussex University, and is currently working on a manuscript about the natural history of microbiology and bacteria. So he's the ideal person to explain to us about the importance of seaweed in microbiology and in agar particularly. So it's great to be speaking to you. So tell us about agar.
Well, I don't know that I'm an authority on seaweed or agar, but I know an agar plate when I see one. So any microbiologist who cultures bacteria will be familiar with the fact that we've been using agar plates as a kind of support matrix for bacterial cultures for over a century now, and the story all goes back to the German microbiology scene in the late 1800s, and Robert Koch, who famously grew the tuberculosis bacterium, was initially he was using things like potato slices as a nutrient medium to grow his bacteria on, and they're not very potato slices aren't terribly good. And then Fanny Hesse, who had been using agar for cooking, said, well, why not use agar? Because it has exactly the right properties that you might want to form a bacterial medium out of that's semi-solid. So basically agar is it's a colloid. It's a chemical substance that's produced by certain seaweeds, and it has the advantage that you can cook it up to high temperatures and combine it with water, and you only need a tiny amount of agar to make quite a large volume of water semi-solid, and it stays semi-solid up to high temperatures. So it has big advantages over other substances that you might think about, like say gelatin that melts, or potato slices that basically sort of break down. And we've been using it ever since. And as simple as that, it worked in 1882 or whenever it was. And until we stop culturing bacteria and go purely over to looking at molecules directly, which I guess is going to happen sooner or later. But until that happens, we're going to carry on using agar because it's just great.
We use vast quantities of it, don't we? There's huge agar farms in Japan and places like that make this seaweed that produces the agar, as I understand.
Yeah, I'm not an authority on this, but it's sort of part of the folklore, part of the lab lore of microbiology, that it was initially sourced from parts of the world where they used agar in the food industry. And Japan is one of those, and it remains one of the major producers. I believe they have seaweed farms where they grow seaweed on ropes to harvest the agar, and I believe there are a few other places around the world where they grow algae to extract agar.
And these seaweed farms are actually carbon negative, as I understand it. So they're actually taking in carbon. So it's one of these kind of agricultural things that's actually very helpful to the environment and might actually help kind of mitigate carbon, like climate change and things like that. Which is
oh, that's interesting. So
yeah.
I hadn't heard of seaweed farms as being being promoted as a carbon sink, but they are, yes.
Yeah, so I was reading about the thing with Fanny Hesse that's quite interesting is that, I mean, a lot of the stories kind of talk about this cooking aspect, but she was actually quite central one member of the lab team as well. So, you know, it was something that she'd been looking into, and then she came up with this idea rather than, because a lot of the mythology around it sort of, and I think I've talked about it myself, is this idea of the cooking thing and the relationship to it. But as I look into it more, it's like, you know, she was one of the scientific team, so she was one of the you know, women's histories sometimes get a bit muddled in science and explained away. So it's quite interesting to bring that kind of aspect forward as well.
Oh, sure. No, she wasn't just a woman in a kitchen. She was, as far as I understand it, she was working in the lab, and is as important as Koch and Petri and all the other people that have probably been.
It's interesting.
Better.
We don't call, you know, we've got Petri dishes and, you know, Koch's posters.
We don't call them
We
Hesse.
don't have a Hesse.
Hesse, a Hesse plate.
Yeah, Hesse plate or a Hesse, Hesse jelly or something. You know, it's interesting that it's not been recognised in that way. Maybe it's something that I can think about, is sort of recognising that story about a bit more in the artwork that I'm doing. But I mean, agar's not just a seaweed jelly now, is it? It's this enormous kind of scientific research area and product in itself. Like, there are so many different kinds of dyes, and I'm always fascinated in the history of dye and microbiology. You know, we have Gram stains for viewing you know, bacteria under the microscope and things like that, and we have dyes that will penetrate bacteria and kill them, like things like Prontosil and the sulfur drugs, which were kind of early precursors to antibiotics. And this relationship of dye and microbiology is always very important, something I've used in our collaborations, because I didn't mention before that you're a very long-term, in fact, probably the longest-term collaborator of mine. We've worked together for almost 20 years on different projects.
Right. That's more important than all the other stuff you've said about me.
Yeah. And things like the MRSA quilt that we made that with James Price as well, that was using growing the cloth onto agar that had dyes in it that revealed MRSA bacteria, so it turned the bacteria blue, so it dyed the cloth blue as well where the bacteria were growing. And so you have these diagnostic agars now that are really quite kind of state of the art, aren't there?
So what's great about agar is that it converts basically water and anything that you want to put in water, like dye or antibiotics or very specific nutrients or whatever, into something that's semi-solid. And indeed, what you do now is that you get commercially produced agar medium that have all the dyes incorporated that you might want for signaling, for example, the conversion of a sugar that's impregnated into the agar into some other substance as a signal for some kind of bacterium that's doing a chemical reaction in it. I mean, when not very long ago, when I started as a trainee in the field, labs were routinely making their own agar plates, and there would be someone in there who was highly skilled, who or even the researchers themselves, like myself, would basically get a big tub of agar and various media, and we'd make up our own agar plates, and there was a certain mystique to it, and it was great fun.
I've done it myself. I've made hundreds of plates with very complicated recipes for streptomyces. It was quite a torturous job I made, because I made a huge amount of streptomyces plates and had to weigh up tiny amounts of these things in quite large quantities to and several batches. It was that was quite an endeavour. So yeah.
Yes, I think it's important to emphasise to any viewers that you have taken on the role of learning how to do lab processes in some detail. You know your way around a lab, and you know how to make an agar plate.
Yeah, I'm very familiar with agar plates. That's why it seemed an interesting thing to try, to do this. What always surprises me, and I don't even know how it works, is that the plates are like pale coloured. They're like usually like yellowy cream coloured, like the ones for the MRSA or something. But when the bacteria touches them, it grows blue. Do you know why?
I did, but off the top of my head, I can't repeat the chemical reaction to you.
Yeah, but it's interesting. So there's some sort of molecular chemical reaction going with the dye in the plate that the bacteria are taking up and then doing something with to make them grow blue. And there's even agars that have multiple colours in. So there's one I've worked with, this brilliant UTI agar for urinary tract infections that I've grown fecal bacteria on with Dr. Nicola Fawcett, and it comes up with pink and blue colours and all sorts of things. It looks really beautiful in a way, which I guess people listening might think is quite a strange thing to say. But you find beauty in all sorts of places. The Victorian seaweed collectors, and it was a very popular hobby with women because Queen Victoria also took it up, and all of these early kind of female seaweed collectors. They called them flowers of the ocean, and so they would have this great appreciation for it. But it was quite strange with me. I was walking on this local beach here for about 6 months of the lockdown before I suddenly at one point said, oh, seaweed's quite interesting and unusual, isn't it? But it was like 6 months that I've been doing this. So yeah, it's a strange thing. Apparently women were allowed to study it because the sexual reproductive cycle of it wasn't very well understood, and that meant that they didn't think they were like sexual beings or something. So the women were allowed to do that, but other kinds of science they weren't allowed. They weren't allowed to study for that reason, in case they were kind of swayed in some way. But
well, they weren't going to be led astray by seaweeds, basically.
No, that was the belief. I mean, maybe it's a similar thing with bacteria, because there were more, and there are more female scientists in areas like microbiology compared to, you know, things like fields like engineering and things like that at the moment. So it's sort of carried on in a way, that kind of thing, to an extent, which is interesting. I'm talking to Jane Freeman and Kerry Davis about this issue and sort of women in science, and how that's developed from Mary Philadelphia Merrifield, our Brighton seaweed pioneer, who got a paper in Nature in the Victorian period, which is quite an achievement. So you said that we might go over to a wholly molecular kind of way of looking at microbes and things instead of using the agar. But I know that there are some scientists who are really dedicated to this idea that you have to be able to grow it as some sort of proof of things, you know, like you have to be able to see it happening in the real world as well. What do you think about that sort of idea of like you have to be able to grow it?
I suppose it depends on what view you take as to how you define what something is. And actually being able to see something growing on a plate puts it into our world. So I guess there's a kind of intuitive or emotional attachment to things that are part of our world, part of our what we consider to be normal experience, as opposed to molecular signals encoded in ones and noughts that give you some idea of the genetic distance of the organism that you've detected versus reference strains of it. So I guess that's more a problem of how humans see themselves and their environment and what's in it than because of any fundamental importance of whether you see an organism in nature or an organism as a molecular signal. I think we could get used to ones and noughts if we look at it long enough.
And you've done a lot of work with genomics, haven't you, with bacteria and whole genome sequencing of bacteria. So genomics is this sort of field of studying the genetic data, the genomes, the whole genomes of
Yeah.
these organisms. And you've done a lot of work with microbiology in that field, and things like
Okay.
Staphylococcus bacteria, for
Well,
instance.
before I mention anything about, before I say anything about that, to give you some background to where I'm coming from in this, I'm actually passionately interested in organisms as organisms, because in my spare time I like to do entomology. So I've spent my whole life going out looking for insects and finding them and bringing them home and identifying them and so on and so forth. And unlike bacteria, you can, by and large, you can examine an insect as an actual animal, and just by looking at it and comparing it with others and measuring it and so on and so forth, you can say what it is. With bacteria, they are morphologically often indistinguishable. They're either round, and we call them cocci, or they're long and straight, and we call them rods or bacilli. And there are millions of species, and how we tell them apart is based on, or it has been based on, what chemical reactions they can perform when they're given various food substances, or what temperature they grow at, or, I mean, they're not part of our world. We can't see the differences between them intuitively or naturally. But if you look at the genetic code of a bacterium, the entire genetic code of a bacterium, and compare the DNA sequences with the bacterium with that of another one, suddenly it becomes obvious what it is and how different it is. So when you look at the world of a bacteria, during the course of my career, for example, it was just starting to become a bit more molecular at the time I started as a trainee. Then access to sequencing technologies became more affordable about, what must be about 12, 13 years ago now, which is why I got so excited to be involved in a project to use that technology. And suddenly it seems like for the first time, they've become visible to us. So the bit of work that you're describing was a research consortium, which was called Modernizing Medical Microbiology. And I was fortunate enough to be part of that from the start. And one of the bugs we looked at was Staph aureus, and we looked at with James Price as a research student, and others looked at the natural history of Staphylococcus in the hospital in Brighton, for example. And by looking at the genetic code of different isolates, you can get an idea of how it's moving around between patients and in the environment, and what it's doing. And we also looked at gonorrhea in Brighton, which is an interesting bacterium to look at, because unlike other bacteria that cause infections in humans, such as Staph aureus, where generally you have quite a lot of information about the history of the patient, with gonorrhea, it's all done rather sort of secretively, because of the legislation, it has to be done in confidence. But if you look at the bacteria, you can form a virtual network of how the bacteria is moving through the human population, just by looking at the genetic differences and seeing how closely related they are. And you can actually build up a world picture of the family history and movement of the gonorrhea bacterium.
And what's interesting about bacteria with genomics, one of the big things is that sometimes species, just from month to month, it seems sometimes that are swapping names, like their taxonomy changes. So like Clostridium difficile is now called Clostridioides difficile, I was informed recently, even though I've been working with it for quite a long time. So it's like because they realize that genetically the connections are slightly different and things like that. And I was really shocked to learn, and because I was talking to a seaweed specialist, Sarah Stewart, I've shocked to learn how little genomic research has gone on with seaweed. She was saying that she, as far as she knows, there's been one whole genome of seaweed sequence. But I mean, I think maybe it's come on a bit since then, but it's obviously very small amount of research in that area. So we have all these different seaweed species that are kind of the names given by the Victorian seaweed collectors. And I can sort of imagine when these all start getting sequenced, we're going to learn a lot more. And there's probably like a whole kind of load of opportunities for synthetic biology in the field, because with bacteria, we have all these kind of like, we've learned how to modify them, and with yeast as well, to use them as kind of factories to produce, you know, molecules or proteins that we want to produce. You know, there's been working with yeast that can produce lactic acid, which can be used to make plastic, PLA plastic, for example. But there are lots of things that they can be used to take in carbon and produce animal feed and things like that. So it'd be really interesting to think about the future where these seaweed and all these organisms that live on them and things, which are probably not that well studied, are starting to be studied more as well. I've been working a bit with Nick Thompson's group at the Wellcome Sanger in Cambridge as well, talking about cholera. And how that sort of lives in the sea. And there are all lots of, so it's not the pandemic strain of cholera, I don't want to frighten anyone, you know, it's not these like deadly disease causing cholera strains, but there are forms of cholera in the seawater, even off the Brighton coast, or species of Vibrio that can even produce light and things that live here. I wonder, apparently there are bioluminescent seaweed as well, I've been told. So yeah, have you got any other sort of closing thoughts on the relationship of agar and seaweed and microbiology and my project?
No, well, just to recap on what you're doing, I believe you're scouring the Brighton coast for seaweeds and doing a total artwork of Brighton seaweed at a sort of lab and molecular level. Is that right? Or
yeah, and also a cultural kind of history of like
cultural
seaweed and in the development of science and women in science, and so tying in a lot of things, this history of agar, the connection to microbiology, you know, the importance of seaweed, this like this forgotten organism, this much ignored organism, and that actually has this huge importance for the field of science. You know, it's not just bacteria we grow on agar, plants, modified plant protoplasts and things like that. We use agar as well. So it's just hugely important for science. So it's a sort of multifaceted view of seaweed, and I've called it Algologies because it's this idea of actually, I mean, this term algology is supposedly it's a misunderstanding. So it's back in the days when they used to think you just put ology on the end of anything, so they put algae for seaweed, algology, but actually the Greek for seaweed is phycos, so it should be phycology, and that's what it's known as today, whereas algology is actually the study of pain. So I thought it was kind of appropriate to kind of think about this relationship during this lockdown period and everything everyone's gone through, being out in nature as well. So that's sort of threading into it. So it's a sort of multi-threaded, entangled, like I guess a rock pool of seaweed. It's entangled project. Yeah.
No, it's great. It's quintessentially Brightonian.
Like the gonorrhea that you were studying.
Seaweed is much more so. I mean, it's of the sea.
Yeah, it's
Gonorrhea doesn't have those Victorian ladies.
Well, maybe.
Maybe I don't know.
Yeah, we haven't got the history of that. So yeah, so Thank you for joining me today and talking about all this amazing stuff and filling me in on all the details of the history of Agar and genomics and this wonderful conversation. So thank you so much, John Paul. Goodbye.